data over 45. We'll just get it back in a minute though. But I want to go through this first. And so immune system, and we are eventually going to get towards respiration. But I want your notes out. Get in that habit of your notes out every day. That's your text notes, your PowerPoints, your study guide questions, your syllabus every time you come in here. So we're going to review the clonal selection process, which is one of the big formal questions. Describe the steps of clonal selection for all three of those groups. Highly recommended to use this diagram. You need to walk through this diagram, look at it carefully, relate it. You got the visuals and you have to relate it to a text description of what's going on. Okay, so think about a sequence. You have to pay special attention to where class 1 proteins are, class 2 proteins. By the way, class 1 and class 2, what does that refer to? What type of molecule? MHC. So what is the M in? How about Amanda? Do you know what the M is? What? Major. Okay. How about the H?
Histocompatibility.
Histocompatibility and the C?
Complex.
Okay, MHC. That's what this refers to. Class 1 MHC, class 2 MHC. So you want to write that down on the sheet. I have described it in the text slide. The other thing you want to recognize the difference between interleukin 1. What color are the dots for interleukin 1? No, interleukin 1. So whatever you need to do on your diagram, if you have it, make sure you understand interleukin 1 is only released in a couple spots here and here. Okay. So you have to realize that's really released by this macrophage. Okay. Interleukin 1 is released by the macrophage. That's why it's a weird shape. What is a macrophage? I actually used it on the biopractical. Monocyte. It doesn't have to be a monocyte, but that's a very common type of, we call it ABC. So that's why it has a weird shape. Remember, monocytes are a little bit oversized and a little misshapen, and they have usually a little notch in the nucleus. But and that's what they're trying to do right here. Okay, so the macrophage ABCs are secreting interleukin 1. And then you see the same thing over here. Pink dots, interleukin 1. What color is the interleukin 2? Yellow. So which are not going to show up if you did a black and white copy. So you need to make sure you realize that the yellow dots are kind of everywhere, but what releases interleukin 2? It says T cells, but specifically helper T cell is the main one, but it could be released by other T cells. But interleukin 2 especially, you want to associate with helper T cell. Here, interleukin 2 released by helper T cell. Okay, so start putting together these things, and I really like this diagram, and I like to coordinate that with the text explanation of the clonal selection of all of these cells. So beyond that, we're still going to get into immune diseases. We'll learn about anaphylactic shock, and we'll talk about histamine again, role of histamine, those for inflammation, allergies, and things like that. And we'll use AIDS, HIV as a big example of immune disease. Okay? And then we jump into respiration. We have the exam coming up a week from today, and I'll talk a little bit more about that. So, use this diagram. Now, just before we get into that, let's review these two. When we get into diseases, I'll get into cancer, anaphylactic shock. But what is an antigen again? How would you describe an antigen? How about a hand up on that? Somebody want to? Christina, any idea what an antigen is? How would you define it? Okay, Okay, a bad guy. That's a starter. Unfortunately, I won't take that on the test. But it is something that might do something bad. But you know, so give me a little bit more. I see sometimes. Yes. Promotes it can. Okay. All right. Yeah. It's a foreign substance in the body. Okay. And so it's basically. Did somebody want to say anything else about that? Yes.
It generates antibody. It's an antibody-generating substance.
Yeah, it's a substance that generates antibodies. So it's any substance. It's usually a foreign substance that stimulates the production of antibodies. Okay, so it could be just about anything. But the key thing is that if it stimulates antibody production, it's an antigen. Okay, so. Substance. Okay, such as, what do I mean by substance? Could be a protein. If it's not a protein, what might it be? I guess it could be a sugar. You could have sugar. There's only proteins, carbohydrates, lipids, nucleic acids. Usually a nucleic acid, DNA, and stuff like that is usually an antibody-generating substance. Sugars, maybe, but generally they're proteins, or it could be just some typical biology molecule. It just could be a random molecule. It could be, I know some people that get sensitized to copy machines, just dust and stuff that they use in copy machines, or it might be a pesticide or herbicide. Or it might be, well, bee venom. Usually venoms from animals and things usually have protein in them, but it doesn't necessarily have to be a protein. It can be a piece of protein. So I'm going to say such as protein or other molecule. Okay, so it's any substance such as protein or other molecule that stimulates antibody production. Now, define antibody, that's maybe a bigger idea than what I want to get into. But first of all, what class of molecule is an antibody? Is it a carbohydrate, a lipid, or protein, a nucleic acid, or none of the above? It's a protein. Okay, so you realize that antibodies are proteins, which means what actually helps code for the creation of a protein? What is the molecule in all organisms that codes for the production of proteins? DNA. DNA. Right? Okay, so that's the whole point behind genes and DNA. So proteins really are the expression of DNA, whether it's your hair, your skin, your muscle, your eye color. I mean, or antibodies. It's another kind of protein. Okay, so it's a protein. And what releases them? What cells release antibodies?
Immunoglobulin.
T cells. The T cells make antibodies or release them. The T cells release antibodies. No. Okay, so killer T cells don't really make them, crank them out. Neither do the helper T cells. So you really need B cells if you want antibodies. So that's super important. So they're released by B cells, and what do they do to antigens? What are 3 ways they can act on antigens?
Clump, coat,
coat, splice. We went through that last time. Clump, which is 2 words, technical words. Okay, how can you clump a molecule? Technical word. Dana Lau, what is the word that means clumping a molecule?
Agglutinate.
Ask Dana Lau. What is the word that means to clump a molecule?
Agglutinating. Did I pronounce it right?
Okay, I'll let you help her out. Did you say it?
Agglutinate, right? Or
Okay, Dana said you agglutinate molecules. Is that correct? What are you going to say, Lisa? What, Lizelle, what? Precipitation of molecules, right? Like chemistry lab. And it's subtle distinction. Basically means the same thing, clumping. They say precipitate molecules. Clumping cells is called, Dana. What? Agglutination. Okay, that's so. I want you to distinguish that because I will ask you this kind of stuff on the test. So you got so precipitate. Okay, so you got a clump. We also have coat. What's the word for coat? Which means to neutralize or the process of neutralization. I'm just going to say neutralize. Or you can lyse. And what technically are the molecules you're going to use to lyse? We have complement fixation, okay? That was the last thing we did. And you're using MAC. What does MAC stand for? Membrane attack complex. So we went through that extensively. You got clump, you have coat, you have lyse. Alright? You need to know precipitate, glutamate, neutralize, complement fixation. Alright? So antigens are the bad guys. Antibodies are the good guys. In a general sense. Very, very, very general sense. Okay, so when we get into this, we're going to have to learn a little bit about cancer, anaphylactic shock, HIV/AIDS, retrovirus, but let's make sure we have all the understanding between the three major categories of cells. There's more than that, but I'm keeping it kind of simple on the test, focusing only on those three based on this diagram. All right, so if we look at, let's see, killer T cell. What's another word for killer? Another word used by the book that means killer. Cytotoxic. What is cyto? Cell. Toxic means it's poison. It's poison to other cells. Now that doesn't sound good, but fortunately it's poisonous to the bad guy cells. So it's going to kill, poison, or destroy somehow the bad guy cells. Now poisoning, it's more like destroying, blowing up. So antigen binds to the APC. Now that's the first thing. So this is the killer T cell. Antigen binds to the APC. What does APC stand for? What is the A in A? Antigen. Which is a substance such as a protein or a molecule, and it's usually a foreign substance. Right? So it's an antigen. P is presenting, C is cell. So it's any cell that will show an antigen to something else. So this is going to be things that like to eat antigens, such as macrophages, such as monocytes. Now you could have other cells, neutrophils, things like that in your body that eat other stuff. So there are different ways to present the antigens, but I'm just going to use as a common example monocytes. And usually we'll just use macrophage, and then when you're in the immune system, they won't even say the word monocyte, they're just going to say APC, antigen presenting cell. So the antigen binds to an APC with a class 1 MHC. Now there's different classes. So what I'm going to have to do is, I want to say, okay, APZ, I'm just going to say presents the antigen. I know I said it fine. So you have an APC that starts off. You don't necessarily have to have an APC in the B cells. And I'm going to say with what class of MHC? And sometimes you use Roman numeral already, like class 1 MHC, right? So make sure you distinguish that. You're going to have to remember which one's class 1, which one's class 2. It's backed up. See, here's the macrophage. It's gobbled up some bacteria. It's processed the antigen, little red spike. The little red spike now is sticking out on this box-shaped protein. That is the class 1 MHC. So the MHC, major histocompatibility complex, is a very important tissue getting along the protein. And what is the main thing about MHCs? What does that help the body do? Not toughness in a general sense. Evan, do you remember what an MHC does? Asra, yeah. Yes.
Doesn't it just present whatever's inside the cell? If the cell's infected, it'll present the antigen. If it's not, it'll just present whatever's in the cell.
Okay, the MHC can do presentation, and in a simple sense, I was trying to get you to say, how do you know if you're in a war and everybody's playing by the rules anyway? How would you know who's on your side and not on your side?
The uniform. The uniform.
Kind of like by the uniform, right? So the MHC proteins are kind of like the uniform. Now you can have different. You can have Army, Navy, Marines. So you can have different types of Marines. Different types of the Army, Navy, Air Force. So you can have like class 1 MHC, class 2 MHC. You don't have to all have the same identical uniform. But the main thing is you have to know, are you on the same side? Self versus non-self. Right? Talk about that. That's a simple way to remember that. Self, that's you, versus non-self, which is not you. If it's not you, then maybe you should not let that in your body. If it is you, leave it alone. Okay? So MHC, the major histocompatibility complex, helps you distinguish yourself or the molecule itself or if it is non-self. So if it's from a bacteria, what would you say? Is that self or non-self? Non-self. Okay. If it's a virus, non-self. If it's a pesticide, non-self. If it's a tree pollen, it makes you sneeze. That's non-self, right? So stuff that gets in your body that's not normally in there. And usually by getting in your body, we usually eventually gets into the bloodstream or into a lymph node or something like that. Okay, so if it just passes through your digestive tract, it might not do anything. Okay, so class 1, I want you to compare that to helper T cells. You use a macrophage or APC in helper T cell activation. See that? Look at that middle column. What do you see? Activation of helper T cells. Are you using a macrophage or an APC there? See this shape right here? What is this cell? That's a macrophage or an APC. That's a macrophage or an APC. It looks like, notice it has an orange shape as opposed to a pink notch. Now that's not literally the shape, but it just gives you the impression that there are different shapes to these proteins. So what class protein is involved here with the activation of helper T cells? Class 2. So what you get this. So you still have an APC presents the antigen. So APCs are very important to starting up this whole process. And okay, W means with class, what class? 2 MHC, right? Whereas killer T cells with class 1 MHC. So it's the same idea. Alright, and alright, so the APC is going to bind to the killer T cell, right? With the class 1 MHC. And I'm just going to simplify, I'm not going to write every step of it. So the APC secretes interleukin 1. And you may recall what usually secretes interleukin 2? B cells. Okay. Or no, not B cells, helper T cells secrete interleukin 2. I lost my Anybody see my Oh, good. Okay, so APC secretes interleukin 1. You also have the helper T cell that secretes interleukin 2. Right, so there's a two-way stimulation here. So maybe I'll just put this up here. You got the APC presents the antigen with class 1 MHC. Okay, so then we have the APC binds to the killer T cell. I'm going to say killer T. And it obviously has the class 1 MHC, so you want to make sure you got that. Class 1 MHC. As opposed to class 2 MHC, right? The macrophage secretes interleukin 1. Which is the APC. Did I use the word I use the word APC. I better stay consistent. But you have to realize that an APC is often a macrophage. So that could be confusing to you if we're both using different terms. APC secretes interleukin 1. I'm just going to use, I better not use it. Okay. So notice that there's direct contact here. Direct contact with the APC. You also have the helper T cell, which secretes interleukin 2. But the helper T cell does not require direct contact. I'm going to say IL2 because I'm running out of space. So this is co-stimulation. You have direct contact by the APC, indirect contact with the helper T cell. And I'm going to contrast that with it over here because you need, with B cell, you need direct contact with the helper T cell. It's not enough to just squirt the spray. You've got to actually connect. Here, it's a little, you don't have to have the helper T cell actually make direct contact as much. Okay? And then ultimately, you get, what are you going to end up with after you clone? You get mature killer T cells, and then what are the ones held in memory reserve? Memory killer T cells. Okay? So then you're going to get, so I'm going to just say IL1 and IL, what does IL stand for? Interleukin 2 stimulates cloning. And of course you may get active killer T's, and then you get memory killer T's. Why is the memory good? Next time you see that same thing, you're going to act a lot faster, and maybe up to 10,000 times more potent in the fight against it. So it's a big deal. Question?
Does the helper T cell have to be activated to secrete IL-2?
Yeah, this is the thing. It's all, it's not linear. So you can't look at only killer T's, you also have to look at helper T's. And I put helper T cells in the middle because helper T cells are kind of like the cracks in the whole system. So, okay, so this is the whole sheet. You all found this on Canvas. And so if you look in the middle, you've got helper T cells. They're doing the same thing. This is all happening simultaneously. It's not like you do killer T cells first, then you do the helper T cells, then you do B cells. It's all going on at the same time. Okay? It just depends on what the antigen encounters first, which one gets stimulated first. They all need to work. It's a three-legged stool. You take one leg away from the stool, the whole thing collapses. All three of these things need to work together. Question?
So does that mean the antigen has to not only activate the T cells, the same antigen has to also activate the B cells and the helper?
Right, the same antigen.
So they have to have the same receptors.
Yes, they have to share the same receptors, which is a good point. So yeah, and that's right. So you notice it's the same shape antigen, stimulating the same, it's also stimulating helper T cell. You can have multiple infections going on at the same time. We're exposed to all kinds of bacteria daily. So you almost never get one antigen in your body at a time. You get thousands of antigens in your body. So most of them you're fighting off fine. So you're not fighting one battle at a time, you're fighting thousands of battles every day and trying to tamp them down. Most of the stuff, though, your body remembers, so it tamps them down really fast, and you don't even know you're sick. You don't even get sick. You don't feel any symptoms. Okay? So, okay, so we have macrophage here stimulates or presents the antigen. That's what I started off with. It had a class 2 protein. And by the way, what about B cells? What B cells? What class MHC? Okay, so we have, you have to have, does this one show an APC presenting the antigen to B cell? So the antigen binds, it's going to say straight up the antigen. Binds to the helper T cell. And what class? Class 2 MHC. So this makes it more like what? A helper or a killer T cell? Oh, I'm sorry. It's binding to the B cell. I was jumping. Now there's a reason why you have that. That it's similar to the helper T cell. Notice that they're the same. Notice that when you go down a little bit further, direct contact. So you have to have the same MHC proteins if you're going to connect. This is why you don't see the direct connection with the helper T cell to the killer T cell. You don't see that direct connection? Why don't they connect? They have different MHCs, okay? So they have different proteins. So you can get the helper T cell with direct contact with T cell. Helper T cell can indirectly stimulate the T cell, the killer T cell, with just the interleukin 1. So it just sprays out the interleukin 1, it drifts throughout the body systemically, it goes into your blood, your lymphatic system, and then that's enough to stimulate this. Along with, you have both interleukin 1 and interleukin 2 for the killer T cell. So on B cells, different. So you got the B cell with the processed antigen, the MHC2. Now it binds directly to the B cell, or helper T cell. This is another way, by the way. I know I have 2 slides for the killer T cell. I have 2 different slides, 2 different ways to describe this. Honestly, there's no one way that you do this. Clicker.
Right here.
I have too many things in my hand. So the, you know, there's no one way to do it, but I don't remember why I put two. I think I figured it was 2 ways. So you got the macrophage with class 1 and patches and presents the antigens and killers and stuff. Macrophage is what secretes interleukin 1. And then what else do you need? What other chemical? Interleukin 2. So it requires co-stimulation. Co-stimulation means both required by interleukin 2, okay, which is secreted by the helper T cells. So the idea is then, again, you still need helper T cells to promote the activation of the killer T cells. You need helper T cells to promote the activation also of the B cells. So these helper T cells are critical to your immune response. And maybe I will jump ahead to HIV. You know what HIV attacks? Helper T cells. So it wipes out your helper T cells. If you wipe out your helper T cells, then you're not going to be able to fully activate your killer T cells. And then you're not going to be able to activate your B cells either. So you're knocking out a really critical leg of the three-legged stool, and everything collapses. And so HIV will kill you because you can't fight off regular diseases. Question?
For cytotoxic T cell activation, you said it has to be a macrophage that the T cell attaches to. Isn't MHC1 on almost all body cells?
Yeah, there's a variety. You know, there's a mix of the MHC1 and MHC2. You're right. Yeah.
Cytotoxic T cells could receive the first signal from any cell, not just macrophage.
Well, killer T cells, though, have the MHC1 protein on their own bodies or their own cells, so that's what they need to interact with. They need to interact with other cells with the MHC1, as opposed to, say, the helper T cell, which has the MHC2. So it's going to interact only with other things that have the MHC2. I'm highly simplifying it, but yeah, that's the general idea. Okay, so then you get mature killer T cells component, and you always have the memory killer T cells. Once everything is activated, all right? And so the killer T cells, shown in this diagram right here, some are round, some shapeshifters, and then this is a pool of muck, which in this case happens to be a cancer cell. Now, and I'm going to be getting into cancer. The interesting thing about cancer is that where does the cancer come from? What are they? From our own body. So would you consider that self or non-self? Self. So this is the dilemma with cancer. Cancer is self. Your body would normally not want to attack itself. That's a good thing. You don't want to attack yourself. So then you have a real tough time attacking cancer. This is why it's very, very difficult to cure cancer. Because it's self-attacking self. And the cancer cells aren't really attacking you so much. It's just that they're misbehaving. So it's really your own cells that grow and grow and grow. And the tumor, they spread, and they usually crowd out the normal functions. And so in some cases, your immune system is able to adjust and realize it's misbehaving, and it may actually secrete some weird proteins that are not normal, and then your immune system can recognize that and attack the cancer and kill it. But in many cases, the proteins aren't different enough for you to recognize, and you won't attack it, and the cancer just grows and grows and grows. It could be lung cancer, breast cancer, brain cancer, and very tough. So, what is cancer? It's your own body cells. Okay. That grow uncontrolled. Okay. Okay, which means that that forms a tumor. And if they grow fast, you get a big tumor. Grow slow, small tumor. There's some tumors that stay very nice and pretty much all together, and then some of them break apart, and that's called metastasis. And if they break apart, they can drift to other parts of your body, and it can spread from organ to organ. And those are the really dangerous. Okay, so here, though, there is hope. Your own body can recognize cancer. Sometimes it needs a little bit of a push to do that. So here's the other thing about the T cells. Where are T cells originally produced? Usually in bone marrow along with everything else. But where do they differentiate? Why do they thymus? Okay. Thymus gland, B cells. Where do they differentiate? Bone marrow or bursa, which is multiple. So you have bone marrow, and fetal liver. There's different ways. These are pluripotent, which are stem cells. Lymphocyte stem cells then they migrate to the thymus where they mature, fully mature T cells, or they can just stay in the bone marrow. This is the whole area is still in bone. T cells stay in bone and they can mature there as well. So T cells need the APCs or infected cells to show them the antigen. That's what this diagram illustrates. So here is the infected cell. These could be pieces of proteins of a virus or bacteria. The MHC molecule looks like a wrench-shaped molecule right here. So what will happen is this complex, and that's why they call it matrix histocompatibility complex, because it also complex with other proteins. It actually processes the antigen and then gets pushed to the surface. So it's like you grab this piece and here's the protein, and I shove my arm through the roof of the building and show everything and say, this is the bad guy, this is what it looks like. Go after it. It could be diamond shaped, square, it could be round. And then the T cell receptor, killer T cell, they use capital T, lowercase c, cytotoxic. That binds. And so those receptors have to be able to match. If they don't match, then there's no activation. So that's why you also need this MHC, class 1 MHC, and the class, and then the T cell receptor matches up. Notice over here, we have a helper T cell. You'll notice they have a class 2 MHC, different shape. They made it square. It's not literally square, which is rounded with the class 1 MHC. But the same idea. So when we say processing antigen, it's a macrophage that is eating the bacteria or a virus. Rip these pieces, take little bits and pieces of the protein. They got many different proteins because, of course, there's many proteins in a single virus. Then they're expressed on the surface, or presented, and then shown through a helper T cell. Now the helper T cell knows what to look for. So that's going to activate the helper T cell. And then the helper T cell will clone itself. And now you have lots and lots of helpers that can go around and stimulate everybody else. So, so when the killer T cell, we say, destroys dangerous cells by lysing them, it's using a little bit different system than what we talked about, complement fixation or MAC. This is actually a little bit more of a general system. So the complement fixation MAC can occur independently of the killer T cell directly attaching. So the killer T cell has its own way of copying holes through. And so you'll notice right here, the infected cell, let's say it's a cancer cell, or maybe it's a cell infected with virus. So, and this is the thing, you actually do have to at some point maybe kill your own cells to clear a disease. Virus may infect the liver cell, and you actually, in fact, have to kill the liver cells that have the virus inside of it. So you have all these little proteins being expressed from the MHC proteins. So the MHC is showing self or not self. Orange is not self. So you need to destroy it. It marks up, matches up with a cytotoxic killer cell. There are a bunch of proteins right here that I'm not going to get really into, but one of them is called the CD8 protein. So they'll call killer T cells CD8, derived from this type of a little anchoring device that helps it lock into place. And once it binds, then it releases proteins called perforin. You know the word perforate? What does that mean? It perforates. Punch holes. That's all it is. Perforates and punch holes. So it does very much the same idea, but just with different proteins as complement fixation. Because that's what MAC, membrane attack complex complement fixation, does. It builds holes. perforin kinds of build holes as well, just a different molecule. And then that allows ions and waters to go in or out. It means that this cell can't control what's going in and out of it, and it essentially dies, it essentially pops it. So that's the idea of lysis. Cell lysis means to destroy or break open the cell. Okay. Now, I'll have to show you some animations later when I can get the computer to behave. So, helper T cells now are very simple. So, helper T cells help activate both the B cells and the T cells. So, for a killer T cell, again, you need direct contact here where it says helper T cell secretes. You need direct contact between a helper T cell and a killer T cell. And they need to go hand to hand, and they need to actually directly connect. They don't. They have different class 2 MHCs, which makes it class 2 versus class 1 MHC. It makes it difficult to connect directly. So then they have to rely on just chemicals being secreted.
Is that what the interleukin is then?
The interleukin is, yeah, the interleukin is the chemical. Those are little dots that I drew on the solid diagram. Okay, so here's the helper T cell. TH is the helper T cell. This is the APC, antigen presenting cell. So what you'll see is it's a monocyte macrophage. It eats up the bacteria, digests it, pulls out little proteins, presents the protein now. Now, if you're going to interact with a helper T cell, what class MHC do you need? Okay, class 2. That's why it appears as class 2, Roman numeral 2, MHC. And so, notice also they call helper T cells CD4. So you'll see that a lot of medical fields or books, they don't even say helper T cell or killer T cell. They just say T4 or T8 or CD4 or CD8, and they're referring to helper or killer T cell. So maybe I should put this up here. So yeah, the helper T cell goes by CD4. Killer T cell goes by what? CD8. Alright. What? You used to call them T4 or T8 as well. Sometimes you say that. Alright, so they bind. Now you have direct contact though. And what this is showing is interleukin 2. It doesn't show any dots here, but this helper T cell actually stimulates itself. That's why they have an arrow coming back or something. It self-stimulates because once the interleukin 2 is released, it actually stimulates all T cells and B cells for that matter. So interleukin 2 causes it to grow faster, clone itself, but it's also going to stimulate the cytotoxic cells to be stimulated as well. So what it's showing here is a helper, TH is a helper T cell. Over here is the cytotoxic or killer T cell. But there's no direct connection. It's just the blue line means the chemicals. Right? And for that matter, you can also remotely stimulate B cells, but B cells to be fully activated, you need direct connection. And so let's go back to this helper T cell activation right here. So we walked through. We've got the APC presents the antigen with the class 2 MHC. The macrophage secretes interleukin 1. So it starts to look kind of like this. The APC, I use the word APC here. I use the word macrophage. Same thing. Maybe to be consistent, I'll use the word APC. So you got the APC binds to the helper T cell. It's the same so far, except that you have different class MHC. Then you've got APC secretes interleukin 1. I'm going to just write IL-1. Okay. Now the helper T cell will also release interleukin 2 to stimulate itself. But, so even though it's itself, still gets the benefit of its own drug. Okay. And then that interleukin 1 and interleukin 2, just like here, it stimulates clonal. Okay, and what do you think you're going to get when you clone? You're going to get active cells, and you're going to get, what else besides the active cells? Memory cells. You always get memory cells. So by active ones are mature helper T cells. I'll just say active. Active helpers. That's it. Yeah, active helper T's. What are active helper T's also called? They expand and they secrete the antibodies. Anybody remember that? Starts with P. I think this is right up there. Oh, actually, no, I'm getting it from my thinking of B cells. All right, no, just go to active helper T cells, and then you get the memory T cells. Yeah, plasma cells are B cells. Okay, so you always get active, you always get memory, whatever it is. Okay? You always have the combination of interleukin 1, okay? Interleukin 1, interleukin 2. On an exam, I'm going to want you to write out the word interleukin. Yes. It should be easy because what does inter mean? Between. What does gluco refer to? Leukocytes, which is white blood cells. So it's a chemical secreted between white blood cells. That's all it is. Alright? So we go B cells. Let's review that one. We've already done this one. So here's the difference. You don't have to have the APC processing at all because you can just directly attach the antigen. So you got the antigen binds directly to the B cell receptor. So you kind of can skip a little step there. And what class MHC do we have? Class 2 MHC. The antigen is processed by the class 2 MHC. The helper T cell, though, binds. That's what's different. Here we have APC binding to all this stuff, but here you have to have direct binding by the helper T cell. Okay? That's an important difference from killer T cells. Killer T cells do not require the direct binding, and in fact, they aren't designed to bind directly with the helper T cell. So the helper T cell secretes interleukin 2 as usual. I'm going to say IL-2, and that's all you really get here because you're not interacting with the macrophage, so you don't get the same benefit of the interleukin 1, but interleukin 2 becomes much more important here because you're making direct contact. The interleukin 2 is released throughout the body, so there is some activation even without the direct contact, but if you want to get the full force of the immune response, you need the direct contact. And so interleukin 2 stimulates the B cell cloning, and okay. Interleukin 2 stimulates cloning. And notice I just have interleukin 2. I didn't really mention the 1, although it doesn't mean it doesn't have any role. This is the most important role. And then you're going to get active B cells. What are active B cells called? Plasma. They inflate and they secrete the antibodies. And I'm going to put a big note there. They secrete antibodies. Okay, because that is important because none of the others do that. That really makes B cells stand apart. You have the active B cells secrete antibodies. And then you also have memory cells. And they just stay in reserve. Okay? So that's just really a review of all of them. And you're going to just need to go back and study them, try to memorize this stuff, look at the diagrams and the charts, try to put it together visually, as well as sort of in a text or written form. Okay? So plasma cells actively secrete the antibodies. They clone. And then the memory B cells are called up to a lifetime. And then we'll have a very fast secondary response. I'm about ready to take a break here. And I just want you to think about all of this stuff. I think if you look at this diagram and you walk through it, that's really helpful. Okay? That puts it all together. Just realize that it's all occurring simultaneously. They all help each other out one way or other. Crux is the middle. Helper T cells, because helper T cells activate everything. And knock out helper T cells, you're going to have trouble, and that's what HIV does. It knocks out your helper T cells. So, if you have any questions, great. Otherwise, I'm going to give you a little break, and then I'm going to start passing back to the lab practice.